Hello everyone. My name is Feng. I'll introduce the paper submitted to iMeta. The title is A Large-Scale Single-Cell Transcriptomic Atlas Indicates the Immune Panorama of Influenza A Infection. This large-scale single-cell atlas of over 612,000 immune cells reveals a fundamental dichotomy in influenza. Mild disease features a protective, monocyte-centric antiviral state, while severe disease is marked by a pathological neutrophil/MDSC-driven hyperinflammatory response. Mechanistically, the CD8/9/12 TLR4/RAGE axis drives a double hit of cytokine storm and immune paralysis, leading to T-cell dysfunction. These findings define key cellular checkpoints and highlight host-directed therapeutic targets. This study has three major highlights as follows. Here is an overview of our study design and the single-cell atlas. We profiled 612,000 cells from 97 individuals across 4 groups. In Figure 1b, this UMAP revealed 44 distinct immune subsets, forming the foundation for dissecting influenza immunity. Next, we looked at how these cells change with disease severity. Clinical severity reshapes the immune landscape. Mild disease was marked by an enrichment of specific monocyte subsets, while severe disease was characterized by a depletion of T and NK cells and an enrichment of B cells. To understand the source of inflammation in severe patients, we calculated cytokine and inflammatory scores for each cell. Scores spike in severe cases. 5 myeloid subsets contribute over 92% of total inflammation, identifying them as key drivers. We then zoomed in on these key subsets. Figure 2CF shows they highly express pro-inflammatory genes like S100A8/9/12, validated by ELISA. Figure 2G maped the dense communication network among these subsets, largely mediated by the S100 TLR4/RAGE axis. CD8 plus T cells follow distinct trajectories. In mild disease, we observed a model of balanced exhaustion that preserved potent cytotoxic function. In contrast, severe disease was characterized by a decline in cytotoxicity and the emergence of dysregulated cellular states. GSEA confirmed that mild cases were enriched for interferon response pathways, whereas severe cases were characterized by apoptosis and mitochondrial bioenergetics. This dichotomy suggested that CD8 plus T cells in severe disease were not merely exhausted but are experiencing a fundamental metabolic crisis. Analysis of CD4 plus T cells revealed that effector Tregs in severe disease underwent a functional switch from immunosuppression to a pro-inflammatory state, characterized by the upregulation of IL-32, S100A4, and HMGB1, thus actively contributing to inflammation. In mild disease, Th2-like cells supported B cells via IL-4/IL-21 and effector CD4 plus T cells mounted antiviral/IFN responses. Conversely, severe disease drove Th2 cells into exhaustion and effector CD4 plus T cells toward apoptosis, mitochondrial stress, and inhibitory receptor upregulation. Severe disease was characterized by an expanded population of antibody-secreting plasma cells with high expression of PRDM1 and XBP1, alongside an isotype shift from IgM in mild cases to IgA in convalescent patients, marking distinct humoral response phases. Unlike mild cases, where B cells showed antiviral pathway enrichment, severe B cells upregulated immunomodulatory genes and regulatory pathways. This indicated a functional uncoupling in severe disease. Antibody production by plasma cells coexists with an immunosuppressive program in their B cell precursors. Finally, we integrated our myeloid findings, which revealed a contrasting functional dichotomy of classical monocytes, a protective antiviral program in mild disease versus a pathogenic shift toward inflammatory mediators and immunosuppressive molecules in severe disease. Severe disease featured CD14 plus HLA-DR-/low monocytic MDSCs with pathogenic gene upregulation, non-classical monocytes losing antiviral function, and immature neutrophils co-expressing inflammatory/immunosuppressive genes, the latter's highest immunosuppressive score implicating them as T-cell paralysis drivers. In conclusion, we have reached the following findings.